So hi guys, welcome to RAAONLINE. So today we are going to see a small session on reasoning out. So it's going to be for MD DNB postgraduate who is going to appear for the university examination, and for theoretical exam we are going to have a particular discussion called reasoning out, where they are going to give the question and we are going to tell the reason why it's going to happen. So we will go into the particular topic now. So the reasoning out number 1, in exponential phase the bacterial growth is progressively slow down. So whenever we are going to see the particular bacterial growth curve, they are going to have something like a lag phase and log phase, which is going to be called as exponential phase, and there is going to be stationary phase and phase of decline. So there is going to be a particular chart, and when we are going to inoculate the particular organism, so there is going to be a lag phase and there is going to be a log phase and there is going to be a stationary phase, and followed that there is going to be a phase of decline. So we are going to have 4 phases. So the first phase is going to be lag, and this is going to be log or exponential phase, and there is going to be stationary phase, and following stationary phase there is going to be the phase of decline. So here there is going to be lag phase, followed by that there is going to be a log or exponential phase, and there is going to be stationary phase, and there is going to be a phase of decline. So here the exponential phase is going to be called as the log phase, and here during the particular log phase or exponential phase there is going to be the depletion of the particular nutrient in the cell culture medium as the particular cell is going to divide. So in case if it is going to be any agar, it's going to be something like an blood agar, or it's going to be a mechanic agar or nutrient agar, it's going to contain the nutrient, and it's going to be any broth like peptone water. When we are going to inoculate it, the particular organism during the particular log phase or exponential phase, they are going to grow and they are going to use a particular nutrient which is going to be present in that particular medium, and they are going to cause depletion of the particular nutrient as a particular cell is going to divide. And as a particular nutrients are going to get depleted, the bacterial multiplication will start slowing down, and there is going to be the progressive slowdown of the bacterial growth curve, which is going to be seen in the particular exponential and log phase towards the particular stationary phase. So during the stationary phase it's going to just maintain, there is not going to be any cell division or there is not going to be any death. So after the particular stationary phase, the particular phase of decline, the particular organism will start dying due to the particular depletion of the nutrient. So in exponential phase the bacterial growth particularly is going to get progressively slowed down. The reason is going to the due to the particular depletion of the nutrient in the particular culture medium. So this is going to be the reasoning out number 1. Now moving on to the reasoning out 2. Moist heat is going to be more lethal than dry heat. In case if it is going to be moist heat, so there is going to be the moisture. So there is going to be the moisture in the particular moist heat, which is going to be a very good conductor of heat, and hence the particular very good conductor is going to cause better penetration and also going to be the lethal activity towards a particular antimicrobial thing. So there is going to be a lethal effect towards a particular bacteria or virus or any fungi, or going to be any enveloped or non-enveloped organism. So there is going to be better penetration of the heat, those particular organism is going to get killed. And where there is a dry heat sterilization, it relies solely on energy transfer of the heated air, and here the moist heat stream works by transferring the energy contained within the heated and pressurized stream, which is going to be called as a saturated steam, and there is going to be better penetration and better killing of the particular organism, and there is going to be better sterilization of the particular object. So this is going to be the reasoning number 2. So moist heat is more lethal than the dry heat, because the particular moisture in the particular heat is going to be a better conductor of the heat, and hence there is going to be a better sterilization efficacy. So this is going to be the reasoning out for moist heat, which is going to be more lethal than that of the dry heat. Now moving on to reasoning out number 3. Live pneumococci R and heat killed pneumococci S, they are going to be fatal when they are going to get injected together. So R is going to be the rough strain, which is going to be non-capsulated, and that's going to be S strain, that's going to be the virulent strain. And in case if it is going to be virulent strain, so this is going to be the particular organism which is going to have a capsule, and this is going to be the only organism and no capsule. So it is going to be live and it's going to be killed. So it's going to follow the particular thing called transformation. So where the particular organism is going to acquire the particular gene, the particular live strain, which is going to be a rough strain and it's going to be non-capsulated and it's going to be non-virulent. They are going to acquire the gene from the dead one, that's going to be the virulent strain, and they are going to acquire the virulent, and it's going to become the virulent strain, and it's going to cause the death of the particular animal, or it's going to be any living organism where it's going to get injected. So both of which is going to get injected is going to be fatal. So here it's going to process come under the transformation. So here the particular cell dies. So cell is going to be the smooth cell is going to die, and there is going to be the spill of genetic material, and this particular genetic material is going to get random uptake by the particular recipient bacteria, and by the random uptake they are going to have the acquisition of the genes from the smooth strain, that is the capsulated strain. So this acquisition is going to be by the rough strain, and they are going to acquire the virulence. Finally, this particular live non-virulent rough strain, they are going to transform into live virulent smooth strain, which is the capsulated strain. So this is going to be the transformation. So here the live pneumococci, which was like a rough strain and non-virulent, they are going to acquire the gene from the dead one, which is going to be in virulent strain, and they are going to acquire the resistance, and they are going to become a particular rough strain, and they are going to harbor that particular virulent strain, and is going to kill the particular mouse. So this is going to be like an experiment called as Griffith experiment, where they have proved using the particular 2 strain, and they are going to use the killed one, and they are going to use the live one, and they are going to demonstrate the particular experiment of transformation by the Griffith experiment. So this is about the reasoning out number 3. So with this, like, we will move on to the reasoning out number 4. So low flour serum slope should not be sterilized by autoclaving. So any culture medium which is going to contain sugar or antibiotics or anything like an egg, we are not going to do the autoclave process. In case if you are going to autoclave, like, there is going to be the decomposition or degradation of the particular amino acid or protein, and there is going to be production of also the toxic byproduct which is going to harm the particular organisms. So here the serum containing medium should not be autoclaved, and not only the serum but also the any culture medium which is going to contain blood, antibiotics and sugar, and we should not autoclave any of the culture medium like Wilson Blair medium or TCBS medium. So it's going to contain sugar, like we are not going to do the autoclave for these. And if these particular things are going to get autoclaved, there is going to be decomposition of this particular material, and there can also be degradation of the particular substance which is going to present in these culture medium, and there is production of toxic byproduct, and again this toxic byproduct, like, they are going to harm our bacteria when we are going to culture it in the laboratory. So this is the reasoning out number 4. Moving on to the reasoning out number 5. So here obligate anaerobe cannot grow in the presence of oxygen. So in the metabolism of energy containing compound, the aerobe will require molecular oxygen as a terminal electron acceptor, and it cannot grow in the absence of the particular molecular oxygen. In case of anaerobe, on the other hand, they cannot grow in the presence of oxygen, and in case if it is going to be an oxygen for anaerobe, it is going to be a toxic substance. So what is going to happen is that there is going to be the reduction of the particular oxygen, which can produce certain products like superoxide ions, which is going to be a free radical, and the particular free radical can go and cause damage to the intracellular level in the particular anaerobic organism, and they are going to produce various free radical species or reactive oxygen species, and these particular reactive oxygen species will not be able to be clumped up, and it cannot be removed from that particular organism. So what is going to happen is that they are not going to be the terminal electron acceptor, so hence it is not going to be the terminal electron acceptor in case of the molecular oxygen. So what is going to happen, like, it is going to get accumulated inside, and there is going to be production of lot amount of reactive oxygen species, and finally the particular anaerobe is going to get killed. So that is the reason why the obligate anaerobe cannot grow in the presence of oxygen. The main reason is that there is going to be the production of free radical such as superoxide dismutase ion, and which is going to cause damage to the anaerobe. So this is about reasoning out number 5. Moving on to the reasoning out number 6. Heterotrope require organic carbon for its growth. So there are like 2 type of carbon. One is going to be the organic carbon, and another is going to be the inorganic carbon. So in case of heterotrope, they are going to utilize only the organic carbon for the metabolism and the growth. So that is the reason why, like, it is not going to use the inorganic carbon. So heterotrope, they require only the organic carbon for its growth. So it's going to have only the usage of the particular organic carbon for its growth. So this is about the various reasoning out, like, which we have seen today. With the upcoming topics in the upcoming session, I will catch you guys later. Till then, Dr. Ashok signing off.